Our next speaker is Dr. Hanadi Rifai from the Civil and Environmental Engineering Department at the University of Houston, and she'll be speaking on the relationship between bacteria and conventional water quality parameters.
Good morning. I'm delighted to be here. It's a great crowd. I want to acknowledge and thank the coordinators of the workshop. One of my favorite topics, so it's pretty special. I want to take you on a whirlwind a little bit. Most of you have a lot of experience with gathering a lot of data, particularly water quality data. And I want to take you through this whole concept that pathogens defy our ability to understand and manage and work with water quality. We have a lot of experience in the Houston metro area and some coastal watershed. I want to share with you some of the lessons learned about this really unique environmental pollutant, the E. coli and the pathogens. So today I'll talk about a little bit some of the conventional water quality parameters that we are commonly used to gathering and analyzing, present some of the data from Houston Metro, share with you some experiments that we've done in the field that are unique and of their own, and then maybe meander, if we have a few more minutes, down to the coast and look at some coastal watersheds and how they might be different than Houston Metro. So starting out with conventional water quality parameters, we won't go through those in details. The ones that are highlighted in red are the ones that we found a lot of good relationships with pathogen contamination associated with those, including total suspended solids, temperature, turbidity, solar radiation, conductivity, and in some ways back to dissolved oxygen, and of course the topic of today is the pathogens. So I'll focus on some of those. Suspended solids, as many of you know, basically increase the turbidity, make the water less clear, and as a result there's this association with the presence of bacteria that like to attach to the particles and then eventually settle to the bottom, present reservoirs of bacteria or pathogens in the sediment. They might grow, they might die. A lot of things are going on in that. Oxygen-demanding material are in some ways also important with regard to the pathogen scenario. Dissolved oxygen is critical to support the life forms that are in that water body, so the more oxygen-demanding materials there are, then they consume the dissolved oxygen and basically cause general degradation of the water quality. As you know, you've been talking about this all morning, the pathogenic organisms. They're of importance in a lot of different ways, and we're here today talking about how they are relevant to the TMDL process and how to understand what they do. And the key message from my presentation today is that they're one of the unique variables, perhaps the other one being dissolved oxygen, that ought to be treated differently when we think about developing TMDLs and doing watershed studies with regard to these particular contaminants. Temperature is very important. We've heard a lot about temperature, whether it's in lab and or in the field, and I'll talk about that and what we know about it relative to pathogen contamination. It affects dissolved oxygen use, and it affects the solubility, the aeration, a lot of the potential interaction within that water body, and microorganisms being one of those entities that use dissolved oxygen. Obviously, you would expect some correlations and relationships there. We'll start with Houston Metro. I would like to acknowledge our partners in the research. Parsons, Mel is here from Parsons. They were with us every step of the way, so I would like to make sure they're acknowledged that they're part of the team. The Houston Metro is a very diverse and big set of watersheds, starting out with Greens Bayou, moving to the core of the city of Houston. You've seen some of the presentations earlier from George about these areas. White Oak Bayou, I'll talk a lot about, Buffalo Bayou, and then down to Braze and Sims, with Greens being sort of, and Halls being the greener watersheds, if you want to think about them that way. And some of these areas are non-tidal. They're still part of the watershed, but because they're non-tidal, they're segregated or treated a little bit differently, as you will see. Looking at the land use, one of the messages I'd like to leave you with today is that the land use has a lot to do with what you see in the water and or the sediment with regard to pathogens. So the more green you see, the less gray and white, which is low intensity and high intensity urban, this is going to be your better water quality than all of those areas that are gray and white because of the development. We'll introduce the ginormous loads during runoff. We'll also perhaps at least in my opinion and my perspective, sort of concentrate the wildlife and change the patterns of how wildlife uses the watershed. So that's one thing to think about. So let's take a look at some of the data from the Houston metro watersheds. And of course, I'm going in the opposite direction. Here is some information about geometric mean for a period of time from 2001 to 2008. So that's every speck of data that's been gathered for E. coli during that point in time. And this is Green's Bayou, which is somewhat greener. The levels here, 0 to 126, that's sort of the standard. And then all the diamonds that are larger than that are going to be somewhat exceedances, as you might understand. Now, these are reservoirs in the Houston area, Addicks Barker, and you'll note that the discharges from the reservoirs are higher than what you would expect, partially because once you start developing the areas behind the reservoir, then you might have little less contact time and not as much settling of the E. coli. So we do have a little bit of a problem with releases from the reservoirs having higher than expected levels. Now, if you recall, White Oak, Buffalo, and Brays are sort of the more heavily developed watersheds, so their diamonds tend to be larger. And as you go into the less urbanized core, then you will see sort of a lessening, but still way above the standard, as you may already have noted. We try to see if there are changes over time, and here I will touch a little bit on the concept that if you look at the early 70s, Houston Metro had a lot worse bacteria. We did the controls on the wastewater effluent, we put permit limits on E. coli, and things improved tremendously. Then we had the dechlorination issue because of the trihalomethanes, and we took out control of the bacteria. There were no bacterial limits in wastewater permits, and because we were dechlorinating to prevent the trihalomethanes, we started seeing perhaps some releases of unacceptable concentrations, not from all wastewater treatment plants, by no means. I don't want to leave you with the impression that that's going, but our experience shows, based on sampling of over 100 plants in the Houston area, that somewhere between 5 to 10% of the plants will actually have trouble maintaining zero or close to zero pathogens in their effluent. And so the idea of looking at that closely and visiting that issue is also worthwhile to do. We don't see any specific trends in time with regard to the E. coli. The one thing is that we do note that some of the watersheds reflect sort of a statistically significant change over time, but most of them really show no trend. And that's because we're looking at a more recent data set from 01 to 08. So when I go back to the 70s, we do see tremendous declines, but then a tapering off and coming up again because of the urbanization and this 5 to 10% issue with the treatment plants not meeting the E. coli zero or or low discharge, basically. So that's kind of an interesting thing. With temperature, we see variable response, at least from field data. Sometimes the E. coli seem to trend downwards with temperatures, sometimes they trend upwards. And the definition here is that a downward triangle means that in general, the geometric means of the cooler days is higher than the warmer days, and the opposite when the triangle is pointing upward. So if you recall, these are all urban, highly urban. They don't reflect the same behavior. So temperature isn't going to be a good indicator per se of what's going on with your pathogens in the watershed. Rainfall, as you might expect, there is a direct correlation. So an upwards triangle means that the runoff days when they happen to sample right after a rain or during a rain, the concentrations were higher, as you might expect. And all of these are, again, in the somewhat more urban parts of the watersheds, and they do reflect that runoff does have a significant effect, as you might expect. So when you look at water quality parameters, which is where we started, we wanted to see, well, if you just looked at water quality, will it tell you what the status of your pathogens in the watershed? We basically determined that it's pretty hard, that there are only few instances at some of the monitoring stations of positive significant correlations between the pathogen level and the water quality variable. More importantly, it was really restricted to temperature. A little bit conductivity, you might see some correlation there. not as much with flow, and I'll touch on that in a little bit. So our typical, or what I call conventional water quality parameters, perhaps are not the best way to think about pathogen contamination. Now, the part that I want to spend a little bit of time on is this high-resolution study that my PhD student just finished, Anu Desai, and did her work in the White Oak Bayou watershed. For a period of 3 months, she did manual sampling twice a day, morning and afternoon, and then after that, she did sampling using automated monitors, and I'll present the data from both of those just to orient you to the watershed. Basically, it's highly urban down here, a little more green up there. Part of the creek itself is earthen, and the other part is concrete lined. That's another difference. It falls out at what we call Heights Boulevard. Brickhouse Gully is one I'm going to show some data about, as I will for Coal Creek. So those three locations are somewhat interesting, and we will refer to those in a moment. Now, this is the 3-month daily sampling where she would go morning and afternoon, basically. And I'm going to show these somewhat complicated graphs and try to go through them. So you've got E. coli flow and TSS on the axes, and here you've got rainfall. So the inverted bars are when it rained throughout the sampling in a 3-month period. These are the temporal from June of 08 all the way to August of 08. So the scale down here will show you that the purple dots, blue line and purple, are the E. coli data that she got. The red is the TSS, and the rainfall, as I said, are the inverted bars. And then you've got the standards are shown in here. The flow is in green and CFS. So kind of a busy slide. Remember, she's doing this twice a day. And I wanted to point out a couple of things about these. These are done at Heights, which is at the mouth of the watershed, and at Coal Creek, one of the tributaries of the watershed. One thing to note is that Coal Creek still has a broad range of, when it peaks after a rain, you still get to the really high concentrations. However, by and large, it's a lot lower when you look at the urban part of the watershed heights. Is at the mouth, you're really, in general, at 10,000. Down here, you're at 1,000. So you do see differences that way within the watershed. I also want to point out that when you do have this high resolution data, and twice a day is relatively high resolution, you will start seeing some direct correlations between, let's say, flow and TSS, but not as much with the E. coli themselves. Take, for example, this period, we didn't have any rain, and there's really not much change in the TSS or the flow, and yet you still got some natural variability going on with those E. coli concentrations. So the point behind that would be that just like in a general population, you might have in a kindergarten class a lot of kids that are the same age, but they don't behave the same. And so E. coli are kind of like that. They're sort of misbehaving children in that bayou. They sort of do whatever they want, and they're not really consistent at all times. We don't really know why, except that they are living forms and they're not, static forms. So that's one thing to think about. Now, when we look at the weekly variability, you will see some interesting trends. Again, I'll keep us to the time and not use up a whole lot of go over. I know we're getting close to the lunch hour. The main point that I will make is that the sampling locations that are further down in the more urban part of the watershed generally had very consistent levels, and they were much higher than the ones that were farther upstream and really in the more rural part of the watershed. So that was kind of interesting to observe. And even on a weekly basis, there's variability in that data when you look at the geomean. So the variability is at the high spatial resolution, temporal resolution, as well as even when you go down to weekly, you're still seeing a lot of that variability. Now, here is our continuous monitoring samplers. These are systems that are about $25,000. You put them out there. We have them such that we can actually, we have what we call trays, and you could do sample collection. We do that on a 30-minute interval, And basically every 6 hours, my students would go retrieve the tray and analyze it for hold time purposes. So it's real time, but it doesn't mean that you can stay in bed and then voila, the computer shows all the results. It doesn't really happen that way. You still have to send the crews out to retrieve the trays. It is sophisticated in the sense that it triggers dry or wet sampling because we've got a rain gauge connected to it, salt telemeter, so we get the standard water quality parameters are sent to the computer at U of H. And so that is really pretty interesting. It's not a technology for your average use. They're very high maintenance systems. They clog up. Houston waters are somewhat turbid at times, and all that sediment really upsets the sondes or the measuring devices. So I'm going to focus on the results from just a couple of points, Coal Creek and Brick House Gully and Heights Boulevard, and show you some of this high resolution data that we've gathered. So this is E. coli at Brick House, which is somewhat of a small tributary. Again, they're a bit of a busy slides. E. coli here, this is a specific event. This is a day, basically, going from 6 a.m. in the morning to late in the day. And I wanted to show you that at the high resolution, the most important message to take from this is do not sample anytime before noon for your E. coli. You're going to get the highest number, so don't do it. You want to sample go from 12 at the latest 6 p.m. You will get the best number. So that's one thing to keep in mind, that they do exhibit a strong, what we call the DO sag. If you recall that when you go measure DO throughout the day, you see somewhat of a similar pattern. So with E. coli, we see the same thing. And it's consistent with temperature. I'll show you that it's also consistent with solar radiation in just a moment. And we saw that for multiple events. I'm showing a lot of graphs to show you that it does happen a lot. And then when the second message to take from that is that a really fine resolution of either 10 minutes or 30 minutes, we studied at 10 minutes and 30 minutes, you don't see as much of the variability. So the variability is when your temporal resolution is too broad. You're looking at a day or a week or a month. The data is all over the map. When you focus and kind of get this high resolution data set, the numbers are somewhat much fairly behaving and consistent. So keep in mind, if you're going to think about this whole concept, we coined the term bacteria diurnal sag, that in the morning you're going to start up high, it'll drop down. For a long period of time, you're going to be below that standard, irrespective of where you are in the watershed. You will meet the standard at some part of the day. And then here is the part that I'd like Dr. Makhtar and his colleagues to perhaps take a look at. Why do we go back up? Is it just because of a temperature, it allows the microorganisms to regrow, or what exactly happens after 6 p.m. that the stuff comes back? In some cases, it comes back to the same morning level. In some cases, it's slower and then peaks back up in the morning. So again, two messages: bacteria diurnal sag, and go out and sample from noon to 6 p.m. That'll give you the best data. We saw that at all of the sites, regardless of whether they're developed or not. This is the Heights, and again, it's the same. The more developed part of the watershed will not achieve the standard all the time, will not achieve the standard for as long of a period of time. So that's one thing to consider, and again, it correlates highly to the temperature. This is the solar radiation, which again we saw that it's a beautiful indicator. So unlike your conventional water quality parameters don't tell you much, solar radiation and temperature are ones that you would want to track at a high resolution. They will tell you somewhat, what's going on with your pathogens in the watershed. So basically, we did that also during storm events, and I'm going to spend a little bit of time on this because there had been some questions and interest. And as usual, pretty complicated slides, but I'll walk you through them. E. coli and flow on the y-axis, time here. This is an actual storm event where you see the inverted rainfall. And basically, you will see attenuation due to this temperature effect that I talked about in the early parts of the day. And as soon as that rainfall hits, you start increasing. And what I want to leave you with the message that looking at turbidity and flow, while when you look globally at conventional water quality parameters that are measured monthly, you don't see any correlations. At this high resolution scale, you do. So, in other words, the turbidity tracks the flow, the E. coli tracks the flow, and this is the actual E. coli pollutograph from which you should be able to estimate your loading that's going in from a runoff event. So when you actually look at high resolution, we do prove that theory, as we teach it in the classroom to all of our students, actually does apply. When you look at a coarse temporal resolution, the monthly sampling and whatnot, we don't see those types of patterns because the data are just all over the map, basically. They're not, behaving in that sense. So again, we did multiple storms to show that this is not just a one-time event. It happens over and over. And we are showing here a compilation of many of the events we did at this one site. We did them at more than one site. And you could see that, in a runoff event, the E. coli loads are relatively, by and large, they're consistent. So in one way, it would be good to do those types of studies to actually quantify exactly what are your runoff flows on this, using this fine resolution. And again, as I mentioned, when you look at in detail the data that we collected, you do see the E. coli and the turbidity tracking really well, even though when you look at the general sampling data, you may not find as strong of a correlation. Now, in the interest of time, I'm going to just kind of fly through a couple of these and show you some important things that I want to leave you with. And this is this whole idea that when we looked at the tidal part of the segments for Metro, again, we don't see the same things. We don't see the same things for two reasons. When you have these tidal segments, you're dealing with multiple, the E. coli and the entero, and there are numerous other complicating factors such as the salinity and then the tide coming in and out of that system. At least in Houston it does for multiple hours throughout the day. So here's some indicator bacteria data and dissolved oxygen information for a lot of these tidal segments. These are 3 different segments, 05, 06, and 07. And again, when you look at bacteria concentrations as a function of the different dissolved oxygen levels that we measure, we don't see much of a difference. In other words, it doesn't seem to directly correlate. So I bet you, though, if we went and looked again at a fine temporal scale, that there would be quite a bit of correlation between E. coli and dissolved oxygen. But from this general monitoring data, you don't see that. Temperature, same thing. TSS and salinity, again, it's just really hard to glean any relationships at this type of scale analysis, at this type of analysis at that scale. And this last one being for tidal Metro, trying to go back to the question, when you have these tidal segments, how do you go through and handle the E. coli, fecal coliform, and entero? The idea being that there is a ratio of the standards. My group has done a lot of work on that, and one of my students demonstrated that for certain parts of the scale, when the E. coli and entero concentrations are relatively low, the ratios of the standards are a very good indicator, but perhaps in a runoff event, that falls apart, that relationship falls apart. So we do need to visit some of those relationships between the E. coli and entero for tidal segments where more than one standard is applicable or is being used at the same time. So I'm going to skip the Dickinson Bayou in general, just show you couple of slides for that. This is the dissolved oxygen in Dickinson Bayou, and that's what we call the spatial dissolved oxygen sag, meaning in the heart of the bayou, that's urban, dissolved oxygen levels tend to be lower. And what I want you to keep in mind is the image, and when we go and look at the E. coli, this is the same part of the watershed that exhibits a low dissolved oxygen, does show the higher E. coli and the higher entero concentrations. So in tidal Metro, we didn't see relationships between dissolved oxygen and fecal coliform and entero, but in the coastal watershed like Dickinson Bayou we did, and we know that the sediment exerts in the bayou a high organic load and so pulls down that dissolved oxygen, and perhaps because of that, the sediment being residuals to all this organic demand, then there's a direct correlation with the E. coli pollution and the entero concentrations in that watershed. So again, Dickinson Bayou is a lot less urban. The urban part of the watershed is this one here that has the high E. coli and the depressed dissolved oxygen. So that was kind of interesting to compare metro tidal to a coastal watershed, and they behave differently. Of course, we see the correlations to salinity and to between entero and E. coli that behaves as you might expect. So to summarize and conclude, maybe give you a chance to ask a couple of questions. As watershed urbanize, you see less correlation between biological water quality and other parameters. We find that when you look at high resolution, and I'm not suggesting that we have to all go out there, put these really expensive monitors and monitor around the clock. What I am suggesting is it would behoove you once in the watershed as you are developing an implementation plan or a TMDL to do some type of high-resolution study, understand how your watershed behaves, and use that information into the future for designing your monitoring and designing your data collection and designing your implementation around some of the things that you see, and possibly repeat that over time as you are implementing controls on your pathogen loads into a watershed. And then finally, at least in my humble opinion as I stand here before you today, we have to start thinking about water quality a little bit differently and not just rely on conventional water quality. And this is just kind of a big broad message for debate that with the pathogens, with things like dissolved oxygen, they ask of us or require us to think about watersheds as living holistic systems that we have to kind of figure out what's going on with them. Particularly, well, we haven't had rain in a long time, but typically we would, and typically runoff would be, an important consideration. So I would urge you to think about that, discuss that as watershed coordinators, and definitely share with us your thoughts on that. So maybe we can look at some other indicators for these watersheds as they urbanize, start thinking about particulate matter and how it contributes to the pathogen problem, and then think about the water itself really being sort of a microcosm where a lot of biological and chemical reactions are ongoing all the time that deserve a little bit of our attention. So with that, thank you for your time, and I know it's lunch, so I wouldn't be offended if you didn't have any questions. You want to get a break, that would be perfectly fine. I would like to leave you with the acknowledgement slide. There's a lot of us that do all this work. HGAC, flood control, helped us with the data, providing access. Texas Commission on Environmental Quality, they funded our research. Many of my master's and PhD students owe their degrees to the Commission, and the EPA funding that comes through the Commission. Houston Endowment has also been a big supporter of our group. NSF GK-12 program, which is a program for PhD students, training them to be good educators and good transferring information, and all of my students, of whom I've listed just a few that have been involved with these projects: Anu Desai, Emil Helfer, Norma Moreno, Yunfei Sun, Stephen Ray, Nathan Howell, Ya Amoa, and Lisa Greco. And then my postdoc, Lakshmana Divagar, who's currently working Rifai group from India, as we say. He's actually remotely working from India at the moment. So thank you so much again.
On your diurnal E. coli samplings, how many different sites did you form that on?
We did 3 sites, basically. We actually did 6, where we did twice a day for 3 months, and then we went down to 2 to do detailed 10-minute and 30-minute sampling using the automated sampler. And we have the ability to move the samplers, to any other site. But in the lifetime of a PhD student, that was pretty much, she did a number of storms and a number of detailed dry weather events.
And those 2 sites, were they on the same bayou? Or?
Yeah, it's all on White Oak Bayou. One was in the tributary, which is the Brick House Gully. The other one is at the mouth of the watershed. Our theory being that, and EPA has written about that, once watersheds start going beyond 50% developed, you really have a hard time controlling that water quality. And even with BMPs, you're still going to have some issues for the pathogens. It just becomes quite a much more complicated situation. So for all of you in the rural watersheds, be thankful. There's a lot you could do there. With urbanizing watersheds, you would need to talk to the BIG, who's doing the bacteria implementation plan. Rachel over there, talk to her.
Question.
Any other questions? Hey.
Those diurnal plots of bacteria were quite stunning. I've never seen anything quite so beautiful as that. I think one of the thoughts that I had, and I would like to give you an opportunity to address it, based on what we saw earlier today with the half-life, roughly a half-life of 2 hours for some of the samples. Is there any possibility that a fixed collection time of sample bottles could have influenced that?
No, we've actually done a lot of QA/QC, and I didn't show a whole lot of that. So, the way it's set up, the tube is flushed prior to the event with a certain amount of water clearing before the collection happens. And we've actually done one-on-one manual and dynamic with the system, and it's very consistent. They're measuring real data. It's not a figment of the setup that you have. And it's not, they're not heated up inside the vials for the 6 hours or being filled with the presence in the vial. So it's really good data. We've questioned that ourselves quite extensively.
I was just wondering, you could produce the same kind of pattern if you collected the vials at, say, 8 a.m. and 6 p.m. every day.
Oh, yeah. I mean, in other words, what we learned from all of that you don't have to do all this in as much of a high beacon at all. That you can get just 2 points, basically, and it would give you, or maybe 3 is what we got it down to. And you can define a curve. You really have to.
One very quick speculation on that trend that you're talking about. Is it possible that as the bacteria die off, the survivors are also, those dead bacteria are the nutrient for the survivors? Is it possible that they are
there's something there, because obviously if you look at effluents, and many of the watersheds will have a plant, discharging at that point, they do discharge and peak flows sort of in that noon by noon, everybody's showering and whatever it is. So there's probably that same pattern in the flows from the effluent. Perhaps slugs of nutrients come through when that temperature kind of cools off a bit, then the opportunity to sort of utilize that and regenerate. We haven't really had the opportunity to pursue that, but that's next.
And very quickly on the TSS, are you going to speciate the TSS for urban versus rural to see organic matter or carbon?
We're really, really very interested in that. I mean, my group to this point believes that it's all in that particular matter and what's going on with it. And we don't have the opportunity with our current projects to do that. We're looking at it in other ways. But that's, again, an excellent area to pursue that. Precisely.
Well, thank you so much, Hanadi. Great presentation.
My pleasure.
Real quickly, somebody left their black sweater at the stakeholder facilitation training yesterday. I think it was a lady from TCEQ, but I can't remember. So if y'all know somebody missing their black sweater, we're going to go ahead and get lunch started. If you'll